चलो मैं थोड़ा सा एक रिव्यू करा देता हूं और अगर क्वेश्चन दिखेगा तो आप मुझसे पूछ लेना। तो हमारे जियोलॉजी के सिलेबस के जेनेटिक्स में सिग्नलिंग मॉलिक्यूल्स इन ह्यूमंस दिया हुआ है। आई personally feel that this is a topic that actually belongs to biochemistry, or for that matter, this topic should ideally belong to cell biology. Nevertheless, wherever it is given, we should be doing it. Now, when we talk about signaling molecules, so by signaling molecules, we mean those low molecular weight molecules which are present in our body and which help one set of cells to communicate with other set of cells. So the cells which initiate the signaling process are the cells which will be synthesizing and which will be releasing the signaling substance. Now, depending on where is the other set of cells located, the signaling substance can pass through the circulatory system, or the signaling substance will pass through the tissue fluid. If the signaling substance is released into your bloodstream, और लेट अस से कि किसी भी एनिमल के अगर सर्कुलेटरी सिस्टम में वो रिलीज होता है, एंड देन इफ द सर्कुलेटरी सिस्टम कैरीज द सिग्नलिंग मॉलिक्यूल from the source cells are those cells which will be basically synthesizing and releasing the signaling substance to the target cell. So that cell will be the endocrine cell, and then the signaling substance will be traveling through the circulatory system to reach the target cell. So this kind of signaling is called endocrine signaling, and all the endocrine hormones are signals of this class. The other group of cells, which are basically the target cells of the signaling event, these cells are characterized by presence of a receptor on their surface. So let's say that this is a cell. So this cell will have receptor on its surface, and the receptor will be very specific to a particular signaling substance, and the receptor will be capable of initiating some sort of downstream process. So this cell will be called the target cell. So in endocrine signaling, the source cell and the target cells are very far from each other, and the movement of the signaling material, the signaling substance, will be requiring the involvement of the circulatory system. Then there is something called paracrine signaling. So in paracrine signaling, what happens? That the signaling substance is released by the source cells in the tissue fluid. There is no involvement of the circulatory system, and the signaling substance will diffuse into the tissue fluid, and by diffusing into the tissue fluid, it will find its target cells. So in this case, what happens that the signaling cells and the target cells, they both happen to be in near vicinity. There is no involvement of the circulatory system. This is known as paracrine signaling. Whole lot of growth factors, they work by way of paracrine signaling. And then there is one more type of signaling agent and one more class of signaling event, that is called contact signaling. So in contact signaling, what happens that the signaling cell and the target cell, they happen to be connected to each other. By connected to each other, we simply mean that they are in physical contact with each other. The best example would be the neurons. So between the neurons, there is signaling by neurotransmitters. The neurotransmitters are released at the axon junctions, and then what we see that the neurotransmitters released by one neuron will be activating the ion channel of other neuron. So these are the three types of signaling events present within any complex animal, and based on this, the signaling agents can be called endocrine, juxtacrine, and paracrine. Juxtacrine is contact signaling, paracrine is nearby signaling, endocrine signaling, where the signaling agent is released in the circulatory system. Now, when we study the signaling molecules in the human beings, so there are two major classifications of signaling molecules. The first one is based on the nature of the signaling molecule. The nature will decide where is the signaling molecule acting from. And the second one is the functional classification of the signaling molecules. So you have seen that based on the nature of signaling molecules, we see that one set of signaling molecule will be hydrophilic. That means it has an affinity to water. It may be charged also in some cases. And this is the signaling molecule which cannot by itself cross the barrier of plasma membrane. So such signaling molecules will be acting on the target cell by acting through cell surface receptors. So these are the signaling agents which act on the cell surface. We also call them extracellular signaling molecules. So in this case, what happens that on the cell surface, there will be a proteinaceous receptor. The signaling molecule will come and bind to the receptor. Now that the receptor is located permanently within the plasma membrane, so the receptor will have to activate some other protein, and in most of the cases, some other protein activates further some other protein, and then there is an activation cascade. At the end of the activation cascade, that protein gets activated which is supposed to be activated. Let us imagine that the last protein that is activated is a transcription factor. So if it is a transcription factor that gets activated, in that case, the last event that takes place in the cell is expression of certain gene. Now the cell will be synthesizing a particular protein or a particular enzyme. And now this enzyme will affect the cell's metabolism. In some cases, the last protein to be activated could be a membrane transporter. In this case, the cell will either be releasing certain substances or the cell will be taking up certain substances. In some cases, the last protein to be activated can be a cellular enzyme. Once it is activated, certain chemical pathways will be taking place within the cell. But in all the cases, in the end, some effector protein will be activated. The effector protein is the protein that actually elicits, that actually causes the cellular response to take place. It can be a transcription factor, it can be an enzyme, it can be an ion channel, or it can be a cytoskeletal element or something that finally causes a target cell to give the response to the signaling cell. But all of this is mediated by what? A signaling agent that by the virtue of its chemical nature, its size, its affinity towards water, it could not have crossed the plasma membrane. As a result, it had to work through a cell surface receptor. That is the first and the major category of signaling molecules. Majority of the signaling molecules that you are going to encounter anywhere in animal biology, they are the signaling molecules which work via extracellular receptor. There is another name for extracellular receptors, and that another name is cell surface receptor. So if you ever come across the word cell surface receptor, do not confuse. It is another name for extracellular receptor. Up until now, whatever you have heard, if you have any question, just raise hand. In front of me, there is a window where I can see all the participants. So if you raise hand, it will catch my attention and I will let you switch on your microphone and ask your question. It is always easier than typing your question in the chat mode. I'm moving further, but you are free to raise your hand. And the moment you raise hand, after completing my sentence or after completing my explanation, I will give you to switch on your microphone. अगर सारी चीजें अच्छे से समझ में आ रही हैं तो it is all right, so far so good. Now we come to the other category of signaling molecules. The other category of signaling molecule is what? Intracellular signaling molecules. Now, these signaling molecules are, to begin with, low molecular weight. They are lipophilic. And since they are lipophilic, they are definitely going to be nonpolar. They are also uncharged. So these are a couple of properties of certain signaling molecules. Example would be all the steroid hormones, then T3 and T4, the thyroid hormones. Then also you have got the example of certain gaseous signaling agents like NO, that is nitric oxide, and second would be ethylene C2H4, not CH4. So nitric oxide is one gaseous signaling agent and ethylene is another gaseous signaling agent. And of course, all the steroid hormones that you know of, and also T3 and T4. So all of these, by the virtue of their physicochemical nature, they can cross the plasma membrane. Therefore, there is no reason why their receptor will be found on the plasma membrane. These signaling agents have their receptors present within the cell. Now, within the cell, where? That's the major question. So within the cell, there are two sublocations. One, the cytoplasm. Two, the nucleoplasm. Cytoplasmic receptors characterize nitric oxide, ethylene, and all the steroid hormones. Their receptors are present within the cytosol. The receptor for thyroid hormones is present within the nucleoplasm. As a matter of fact, it is already bound to certain gene. Only thing is that when the thyroid hormone comes inside of a cell, then what it will do, that it will go inside a nucleus. If T4 enters the cell, so T4 will convert into T3 and then T3 will get into the nucleus. And within the nucleus, the receptor that is called TR, thyroid receptor. So TR is already bound to the promoter of a specific gene. When thyroid hormone binds to it, the receptor will become active as a transcription activator and the target gene will be activated. In a similar manner, when we see the steroid hormone receptors, even though they are located in the cytoplasm, these receptors, when they bind to the steroid hormone, the receptor hormone complex will migrate into the nucleus. And then it will be binding to a specific target sequence of a specific gene and it will be activating gene transcription. You can more or less conclude that all the steroid hormone receptors and all the thyroid hormone receptors are essentially transcription factors. They are waiting for the hormone to come and bind to them so that they can act to activate, in some cases, repress the transcription of certain target genes. But the same cannot be said about nitric oxide and ethylene receptors. Majority of the time, these receptors are enzymes. They will go and act upon another set of enzymes, and by this interaction with another set of enzymes, they will be activating the effector enzymes. But so far as steroid and thyroid hormones are concerned, their receptors are transcription factors. So take a look here. Steroid hormone crosses the plasma membrane, comes in the cytosol. This is the receptor, and this complex is hormone receptor complex. It moves into the nucleus, and then the hormone receptor complex binds to a certain gene in the region which will be making it activate the transcription. And then it causes transcription of a specific gene, and that way it will be triggering gene expression. And that causes the cellular response to take place. I hope you have understood this thing. So all the endocrine hormones, now we are coming to one level next. All the endocrine hormones which come inside of a cell are called class 1 or type 1 hormones. And all the endocrine hormones which act through the extracellular receptors are called class 2 hormones. So now we come to the functional types of signaling molecules. So first and foremost, hormones. So hormones are basically signaling agents, but one thing that makes them unique, that they are released by a specific glands, and when they are released into the bloodstream. If it is a class 1 hormone, it is going to be lipophilic, and when it is released into the bloodstream, it will need a plasma protein to bind and help it in its transportation. By being lipophilic, it will not be by itself moving into the aqueous medium that is forming the foundation of blood. So it will be needing a plasma protein. But if it is a type 2 hormone, it is already hydrophilic. So type 2 hormones are not going to need any plasma protein. For example, insulin, glucagon, human growth hormone, they are not going to need any plasma protein. They will be moving in the bloodstream entirely by themselves. Eventually, the type 1 hormones at the target organs will be dissociating with their plasma proteins, and then they will be entering into the cell. On the other hand, type 2 hormones, they are not bound with any plasma protein, and on the surface of the cell, they will be meeting and they will be binding to their receptor. Target cells are those which have got the receptor, and the cells who do not have the receptor are not the target cells. So I hope up until now we are clear about what the hormones do. So hormones you have already studied in physiology. So hormones are regulators of various bodily functions, and together the hormones help the body achieve homeostasis. So we are not going further into the hormones, neither are you expected to go deep into hormones within a chapter on signaling agents. Functionally, the second and big class of signaling agents are neurotransmitters. So they are the chemical messengers which are released from neurons and act on other neurons or target cells. Mostly the target cells other than neurons happen to be the muscle cells, which receive these neurotransmitters, and the muscle cells get excited. So at neuromuscular junction, there are specific neurotransmitters which act upon non-neuron targets. Okay, and then they convey signals and regulate the activity of the nervous system. Lot of example: dopamine, serotonin, acetylcholine, gamma aminobutyric acid, so on and so forth. So what they do, they bind to the receptor of the target cell on the cell surface. And all these receptors, which are the receptor for neurotransmitters, are ion channel linked receptors. So to say, when these receptors bind to the neurotransmitter, the ion channel associated with these receptors will become active, and the neurotransmitter will cause a specific type of ionic flux across the membrane of the neuron that causes action potential to traverse through the nervous system. Next one, so this is the mechanism with which you are already familiar with by the virtue of having studied it in physiology. Next one is cytokines. Cytokines are again cell surface acting signaling molecules. They do not act on the receptors which are present inside of the cell. They are proteins, and therefore their receptors are located on the cell surface. These are the proteins which are released by the immune cells of the body. And these proteins control the activity of other immune cells. So basically, the cytokines are supposed to create a concerted immune action, that if there is a pathogen and a lot of immune cells need to be activated for a proper response to that particular pathogen, in that case, cytokines will be activating all these different types of immune cells. But a moderate amount of cytokine is enough to achieve that. If there is excessive amount of cytokine, in that case, the immune system will be so much triggered that it can forget the distinction between self versus non-self. And in that case, the immune cells can start destroying the body's own cell. I'm mentioning this because in acutely serious cases of COVID, there was a phenomenon known as cytokine storm that will hyperactivate the immune system, and the immune system cells would start destroying the body's own cell. And this cytokine storm converted a normal COVID infection into a serious one, and it causes mortality in so many cases. When your immune system is hyperactivated, the tendency of clotting will also be very, very high. And a lot of COVID mortality happened because of the thrombotic condition, that somewhere deep within the body, blood clot forms and it obstructs the flow of the blood. Nevertheless, under normal physiological condition, cytokine is something that will be regulating the activity of your immune cells. Finally, growth factors and death factors. Growth factors and death factors are the factors which will be controlling the division of the cell, maintenance of the cell. On the other hand, death factors are the proteinaceous factors which will essentially be causing programmed cellular death of the cell, which is also known as apoptosis. So, growth factors will cause either maintenance of the body cells, and majority of the growth factors, they act as mitogens. What do we mean by mitogen? We mean by mitogen the ability of a substance to cause mitosis. For example, epidermal growth factor actually causes epidermal cells to divide. Similarly, there are a lot of growth factors for different parts of the body, especially during early stages of animal development. On the other hand, there are death factors. The death factors are essentially those signaling factors. When they target a cell, they will cause very clean elimination of that cell. What do we mean by clean elimination of the cell? We mean that by apoptotic pathway, that cell will be step by step destructed and it will eventually be eaten away. First, the cell will go through a breakdown, and then small fragments that arises from this broken down cell will be eaten away by nearby neutrophils and other macrophages which are present within the tissue. So that is death factors. One of the best known death factor is TNF, that is tumor necrosis factor. So the cells which tend to divide unnecessarily, they are eliminated by your body by tumor necrosis factor. If tumor necrosis factor does not work, then a lot of cells can divide unnecessarily and can cause tumor. A non-benign, that is malignant tumor, is another name for cancers. So programmed cellular death is again a very, very important process, and that is taken over by the opposite of growth factors, that is the death factors. So in this answer, which is going to be in response to a direct question, यहाँ पे questions are not going to be twisted. If ever you come across a short note on neurotransmitter, please understand that the short note is not from the topic signaling molecule, the short note is actually from another topic, and that another topic happens to be what? That another topic happens to be neurotransmitter, as it is specifically mentioned in our animal physiology syllabus. So that is all about this topic. Nothing more needs to be done. If you have still any question, you can raise hand and I will be taking those questions. And now a quick review of proteomics. I suppose कि वो मेरे लेक्चर को आपने देखा ही होगा, but चलो, because you are not asking question, so I am giving you a quick recap of proteomics. First thing first, there is going to be a direct question on proteomics. At the most, there will be a comparison with genomics. So now that you know about genomics, you should be preparing a short answer on differentiating genomics and proteomics. Having said that, proteomics is basically the study of proteome, and proteome is basically the sum total of all the types of proteins which are present in a given cell. A cell contains the entire genome, but all the genes of the genome are not necessarily expressed. For that matter, within the human body, there is not a single cell that expresses all the genes which are present. There are specific genes which are expressed according to the differentiation of the cell. So the liver cells will be expressing different set of genes. Epithelial cells of intestine will be expressing different set of genes. Gonad cells will be expressing different cells of different genes of the genome, so on. Okay. So when you talk about a proteome, so you basically do not talk about the entire organism. What you talk about is the cell's proteome. So by studying the cell's proteome, you get to know what all proteins are there. That way you can infer what all genes are expressing, and then you can correlate that to produce this kind of differentiated cell, these all genes are necessary. And you can get to understand the basis of cellular differentiation. Not only that, in a lot of diseased cells, especially the cancerous cells or hyperactivated immune cells, which lead to systemic inflammation and whole lot of diseases, especially autoimmune conditions. So in all these conditions, studying the proteome will give you a sense what is wrong with the cell, and that will help you devise treatment at a very, very precise level. It could be therapeutic drugs, or it could be gene therapy, or it could be any other approach. But knowing the proteome of the cell is a very, very valuable exercise. In this, you are going to mention certain techniques. So first and foremost, there is gel electrophoresis. So whenever we talk about gel electrophoresis, so we are basically talking about gel that is present in a channel, and then there are electrodes. So when you switch on the electrodes, so based on charge, the proteins, or for that matter, using the gel, you can separate DNA and RNA as well. So when you run the gel, so depending on charge, the protein will start moving to the opposite electrode. Now, the protein which is the smaller will be able to move faster through the gel, because gel offers resistance to the passage. But if a protein is a smaller, it will be able to find its way through the gelatinous matrix much more easily compared to the protein that is larger. Same principle applies at the level of DNA also. That when you are talking about DNA, so a DNA that is longer will be traveling slowly on the gel, and the DNA that is shorter will be traveling faster on the gel, right? So gel electrophoresis helps you separate the proteins. So that is the quality of gel electrophoresis. And once you have separated the complex mixture of proteins, so based on the expected molecular mass, you can identify the protein that you are looking for. So that is one. Then there is spectrometry. So when we talk about spectrometry, so spectrometry identifies and quantifies the proteins based on their mass. Spectrometry is a pretty sensitive process, and in spectrometry, we actually go for MALDI-TOF spectrometry, right? So MALDI is matrix assisted laser desorption ionization. So in this उस लेक्चर में मैंने एक छोटा सा डायग्राम भी बनाया था कि in a metrical material the proteins are mixed, so that using laser we can ionize the proteins and then we will try to separate them. And based on that, we will be trying to identify the protein that we are looking for. Now, there is something called two-dimensional gel electrophoresis. So this two-dimensional gel electrophoresis is essentially the combination of gel electrophoresis and mass spectrometry. तो ये प्रोटीन्स को सेपरेट भी करता है और आइडेंटिफाई भी करता है. By separating, we mean that it can analyze a complex mixture. It can separate the components of a complex mixture. Then we are at applications, something that we have already spoken about. So उसको repeat करने की बिल्कुल भी जरूरत नहीं है. If there is any question, so I am taking a quick pause here, and if there is any question, अब आप लोग एक बार पूछ लो, and then we will be moving further. ओके, चलो फिर हम लोग जो एक फ्रेश टॉपिक है उसको कर लेते हैं, एंड देन विल टॉक अबाउट द क्लास टाइमिंग एंड ऑल. ओके, सो हियर वी आर. So now we are going to discuss a topic called signaling pathway defects and their consequences. So there are a lot of vital roles which are played by signaling molecules and their signaling pathways. Like signaling molecules can act as neurotransmitters, growth factors, death factors, cytokines, and above all as hormones. So in so many cases, we see the signaling pathway getting error. There can be so many points of error. For example, because of old age, the endocrine system is not working well. Now there is less amount of signaling factor. Due to some autoimmune condition, the gland has been destroyed. And because of that, the signaling factor is much less. What could be your example? Your example is Hashimoto's disease, in which the thyroid gland is destroyed by an autoimmune condition. And second is type 1 diabetes, in which autoimmune condition causes the destruction of pancreatic beta cells. And because of that, the secretion of insulin is almost nil. These are two conditions where, because of autoimmune condition, the hormone itself is not being released. Then we can see gene mutation taking place, because of which the receptor may not be present. Or for that matter, gene mutation can alter the behavior of the receptor. For example, in a lot of cancer causing scenarios, what happens that a gene mutation gives rise to an altered form. Gene mutation gives rise to an altered form of receptor. And this is such a receptor which was originally supposed to respond to a growth factor. And growth factor mostly acts as a mitogen. That will mean that if it is a growth factor, it will stimulate mitosis in the target cell. So how will the growth factor act upon the target cell? Growth factor will be acting upon the target cell via a receptor. What if the receptor is being produced by a mutated copy of the gene, and the nature of the receptor is changed in such a manner that receptor does not require the growth factor to get active? In that case, the receptor will always be active, and the receptor will always be triggering cell division. And in this case, the result will be cancer. So what we see, mutation in the gene that synthesizes the receptor. Similarly, mutation in the gene that will be causing the synthesis of some transduction pathway protein. In signaling molecules, we have seen that the signaling molecules that work via the cell surface receptor, they work through a number of intermediary proteins, which we know as the signal transducer proteins. So if any of the signal transducer is aberrant in its behavior because of mutation in the gene that was supposed to encode it, then the signaling pathway will get error. And when the signaling pathway gets error, depending on what is the signaling process related to, how crucial is it for cell survival, if it is leading to less level of signaling or much higher level of signaling or totally a different type of signaling, the consequence will vary. In this chapter, what you should be learning about, and again, this is a topic that gives you a straight question, describe the defects in signaling pathways and their consequences. So you should be knowing what is meant by defect in signaling pathway, what are the underlying factors behind the defects, and then finally you should be substantiating it with certain real world, certain real life examples. At least 4 examples with a little amount of description would be highly appreciated. There can be many more examples, but there is always a limitation on how much you can write for a short note, or for that matter, even a 15 marker, how much can you write? Therefore, we can restrict ourselves to 4 examples. Although in this chapter, I have compiled 5 examples. I can give you more examples, but that will be futile. So here we are. Now let us see what are the concepts. So first and foremost, we are setting the background that signaling pathways, they govern cell-to-cell communication within the body. And they are basically a series of molecular interactions which transmit information from the cell surface to cell interior and elicit a specific responses. So most of the time, this is how signaling process takes place, right? Yes, certain signaling agents get inside of a cell and all, but most of the time, the signaling agents, they act via cell surface receptor. And whole lot of cellular processes, they depend upon the signaling pathway. There is no two way about it. Cell growth, differentiation, survival, and not only that, metabolism and all of that, right? So मैं mention कर ही देता हूँ, survival यहाँ पे लिख देता हूँ, metabolism, so on. So if there is a defect, then it can have a significant impact on normal cell function. So that basically means that depending on which signaling process was it and how did the signaling process get impacted, that means less of the signaling process, more of the signaling process, or totally an aberrant line of signaling process. Let's imagine less of signaling process. Then the body will suffer the deficiency of certain very important substance or certain very important metabolic process. This is what happens in Hashimoto's disease. This is what happens in type 1 diabetes. Very high level of signaling process. In this case, what happens? We see a lot of cancers happening. That if the signaling agent and its receptor would have been working normally, it would have basically given rise to normal level of cell division. But because it is aberrant and it is showing a very high level of expression, so the cells will be dividing unnecessarily. And this unnecessary aggressive division of cells is the underlying cause for cancer. In some cases, rarely that happens, that signaling process will go in a different direction altogether. So these are all defects in signaling pathway. I would dictate something and you please write the proper definition of defect in signaling process. So I'm just dictating and you please write it down. Defects in signaling pathway refer to any unusual deviation in the normal process of cell signaling. It can arise due to changes in the supply of signaling substance, or changes in the availability or functioning of the signal receptor or any downstream signal transducer. The defects in signaling process lead to any one of the following three. A. Less than required levels of signal response pathway, which can go up to total absence of response. B. Higher than required levels of signal response. C. An altogether different type of signal response. Change the paragraph. Such defects arise from physiological factors, old age, genetic mutations, certain types of infections, and exposure to certain chemical substances. तो ये एक हो गया होलसम डेफिनेशन हमारा. If you want to open your answer with this entire definition tiny paragraph, feel welcome to do that, because most of your competitors are not going to get it in any book. For that matter, I also did not get it in any book, and there is no book in Cell Bio that has this chapter called Defects in Signaling Pathway. So to get a sense of defects in signaling pathway, you have to go through the books of medical pathology, where you start getting these defects, and then you build a chapter to teach or a chapter to write these answers. So on your behalf, I have done this. Now, the major types of defect. So there can be receptor defect. That will mean that there is some problem with the receptor. Right from receptor being totally absent to receptor being mutated, so on. Second is signal transduction defect. Receptor is fine, but somewhere the downstream signal transducers are not present, not normal or something. Then transcription factor defects. When we mean transcription factor defects, we mean such signaling pathways in which at the end there is some transcription factor getting activated, and this transcription factor will cause the expression of certain target gene, and the expression of this target gene is going to be cellular response. But what if everything is normal, the signaling agent is present, the receptor is present, the signal transducers are present, but in the end the effector, that was the signaling triggered transcription factor, what if that is mutated or that is defective? Then the entire process that took place before this goes down the drain. Next one is target gene defect. Let's suppose everything is fine, but what if the target gene's promoter itself is defective and it would not be responding to the transcription factor that is at the end activated? That does happen. Moving next, feedback regulation defect. Any signaling process after a certain level of cellular response needs to shut down so that the cell does not respond excessively. What if the feedback regulation process is itself defective? In such cases, the cell will be giving much higher level of response than what is required. Presence of external substances. So external substances can be agonists. So agonists can bind to the receptors and mimic the action of the natural ligand, leading to overactivation of the pathway. So these are hormone analogs and all. If they get into your body, so they will be activating the receptor even without the presence of the hormone. So that is agonist. Then there are antagonists. They can bind to the receptor and block the action of a natural ligand, and that ways they lead to inhibition of the pathway. In some cases, not exactly the receptor, but some transducer or some downstream activity can be blocked. So what happens when you drink coffee? So I have not included it as a defect, but what happens when you drink coffee is that the receptor and signal transduction process that is activated by epinephrine. So in the process, cyclic AMP is generated. Now cyclic AMP will activate breakdown of glycogen into glucose, and for a short burst of time within your body there will be higher blood sugar level, high blood glucose level, and that ways you will feel much more energetic after initial consumption of coffee, but that is only for a certain time. Besides, because it also activates other epinephrine targets, so you will feel more alert, more focused, everything. So what coffee does, that in this signaling pathway, the cyclic AMP that is generated, so cyclic AMP is also eventually degraded. But the enzyme that degrades cyclic AMP, that enzyme can be blocked by caffeine. And because that enzyme gets blocked by caffeine, so it so happens that the cells start showing higher level of cyclic AMP accumulation. And as a result, for quite some time, you will stay alert, your body will show higher blood sugar and so on, right? So that is what a classic antagonist. And there are a lot of pharmaceutical drugs which are classic antagonists, okay? Now, what are the underlying causes of defects in signaling pathways. We have spoken genetic mutation, ये रिसेप्टर जीन का हो सकता है, ये सिग्नलिंग फैक्टर अगर प्रोटीन है तो उसकी जीन म्यूटेट हो सकती है, any transducers gene can be mutated. Epigenetic changes, that there is no mutation, but what if the gene gets methylated and the gene becomes non-expressional? There are a lot of environmental factors like exposure to certain toxins, certain radiation, and all of that. Then diseases and infections. So, like I said, autoimmune diseases can destroy the endocrine cells of a given gland. Similarly, certain types of infections. So, when oncoviruses infect a person, so oncoviruses will alter a signaling process in a manner that the cell division becomes uncontrollable. So that is as that happens as a result of oncoviral infection. Then of course with aging, a lot of signaling factors, they start synthesizing, they start getting secreted less and less. And as a result, we see the signaling process is also getting adversely impacted. Then some defects are because of certain types of environmental exposure or lifestyle choices. So body cells become less sensitive to insulin. Insulin is the hormone that triggers glucose uptake by body cells. So as long as the body cells are sensitive to insulin, your blood glucose level will be within the normal range, because every time you consume food and your blood has got an elevated level of glucose, it so happens that insulin will cause whole lot of cells to take up that glucose, and then the cells will be using that glucose. But if over a period of time the person is feeding much more than what the person should be feeding, and the person has got a good amount of adipose tissue, especially visceral fat accumulation, it so happens that a lot of body cells become much less sensitive to insulin. How did it happen? It happened because of lifestyle choices, not environmental exposure. So that is the classic type 2 diabetes, right? And then we see the defects, the examples of defects in signaling pathway. So the elaboration is there. Let's focus on 4, maximum 5 of these examples. So first example is growth hormone signaling pathway. So growth hormone signaling results in the activation of transcription factor STAT5, and that causes cell growth and differentiation. If the growth factor receptor is mutated, then it is non-functional and it leads to growth disorders such as dwarfism. So here what we see is the receptor gene getting mutated. Because of that, the receptor became non-functional. Then insulin signaling pathway, something that I was telling you, that insulin binds to the insulin receptor on the cell surface. So the receptor is on the cell surface and it activates what? Intracellular signaling molecules that lead to uptake and utilization of glucose. And defect in insulin receptor, such as the one that renders it non-functional, leads to diabetes, or for that matter, lack of insulin sensitivity also leads to diabetes type 2, right? Then we come to TGF beta signaling pathway. So what is TGF beta protein doing? It is also involved in cell growth and differentiation. So normal TGF beta binds to its receptor on the cell surface and it activates intracellular signaling molecule. It regulates the cell cycle, and depending on what kind of level, what kind of receptor was involved, it can either promote or inhibit cell division. So what happens? Defect in this pathway can lead to cancer because it disrupts the normal control over cell cycle. Then there is Wnt signaling pathway, WNT, but it is pronounced as Wnt. So this is also involved in cell multiplication, that is cell proliferation. So this also regulates cell cycle and promote or inhibit cell proliferation. Pathway is different, but eventually the larger objective is same as TGF beta. So this defect also causes cancer because it disrupts the normal control over cell signaling. Then there is hedgehog signaling pathway involved in cell differentiation. This also leads to cancer, and this regulates differentiation of the cell. So what happens? That when there is defect in hedgehog pathway, the cells, rather than getting differentiated, the cells get into the mode of multiplication. As the cell gets into the mode of multiplication, obviously it will be leading to cancer. Finally, one example of external biological substance, that is cholera toxin, that is secreted by Vibrio cholerae, and this is a proteinaceous substance which interferes with the normal cell signaling pathway. So what it does, cholera toxin binds to and activates the G protein coupled receptor known as GM1 ganglioside receptor, and it causes inhibition of chloride channels leading to osmotic diarrhea and dehydration. So the cell will be oozing out water, and this water will eventually be coming out of the body through the dysentery or through vomiting. So this is what cholera toxin does by disrupting G protein mediated signaling pathway. So that is about the signaling pathway, and this is your conclusion. And one good thing about studying the defects in the signaling pathway is that it helps us understand the cellular mechanism of certain diseases, and it allows us to treat certain diseases with signal agonists or signal antagonists. If there is much higher level of expression that needs to be controlled, and if there is a clear and safe antagonist available, this will be a good pharmaceutical drug of choice. And in the other case, where the signaling substance is less, then agonist or the signaling agent itself can be supplied, can be provided to the patient. For example, both type 1 and type 2 diabetics, they have to take insulin. In type 2 diabetes, when insulin resistance increases beyond a threshold level, then it is given. But in type 1 diabetes, insulin injections, they are very much required. Same goes for defects in thyroid hormone. So it could be physiological or it could be autoimmune. But if there is less level of thyroid hormone secretion, then what do we see? We see that thyroid supplements are given to the patients. So this was your chapter on signaling pathway defects and their consequences. Define, give an introductory picture, talk about what all kind of defects can be there, what are the underlying causes, and then substantiate it with 4 or 5 examples depending on the length of the answer. So that is all in this topic. And the only question that you should be preparing, outline signaling pathway defects, explaining its underlying causes. Give suitable examples to illustrate this phenomenon. So that will be the question for which you should be writing the answer. This handout alone is going to be sufficient.